Hello and welcome. As a cardiologist, I've spent my entire career managing heart failure. We've got some great therapies, don't get me wrong, things that can slow the disease down. But what we've always lacked is the ability to truly repair a damaged heart. Today, we're going to take a really close look at a field that promises to change all of that: cell therapy. We're going to dive deep into a landmark analysis to see if we're finally on the verge of regenerating this incredible organ.
This quote really just cuts right to the chase. Ischemic heart failure isn't some small, niche problem. It's a massive global crisis, and it's growing every single day. As our population gets older, more and more people are surviving heart attacks, which is great, but they're often left with hearts that are permanently damaged. Our best medicines are good, but they just aren't enough. So you see, this isn't just some abstract scientific problem. This is a real, urgent human need. We are desperately looking for something that goes beyond just managing symptoms, something that offers a real chance for repair.
So here's how we're going to break this down. First, we'll establish the problem, the unmet need. Then we'll look at the promise of regeneration. After that, we'll put the hard evidence under the microscope. We're going to ask the two questions every doctor asks. Is it safe? And does it work? Before we try to zero in on the ideal patient. And finally, we'll give our verdict on what the future of heart repair might look like.
Now, before we can really appreciate a potential solution, we have to fully grasp the problem. To really get why this matters, we have to understand what our patients are up against. After a heart attack, a part of their heart muscle actually dies. It's gone, and it gets replaced by scar tissue. The thing about scar tissue is it doesn't squeeze, it doesn't pump. This permanently weakens the heart, and while our medications can help the rest of the heart work a little better, they can't bring that dead tissue back to life.
The specific condition we're talking about is called ischemic heart failure, with reduced ejection fraction, or HFrEF for short. Now, when we talk about ejection fraction, all that really means is the heart's squeeze power. It's the percentage of blood the heart pushes out with every single beat. A healthy heart, it's got a squeeze power of maybe 55 to 65%. In HFrEF, after that heart attack damage, that squeeze power plummets, often way down below 40%. And this, right here, is the fundamental problem. Unlike your skin, which can heal from a cut, or your liver, which has amazing regenerative powers, the adult heart, well, it just can't really fix itself. Once those heart muscle cells, the cardiomyocytes, are gone, they're gone for good. The heart just patches over the damage with a scar. And that's exactly why heart failure is this chronic, progressive disease. The rest of the heart tries to compensate for that weak spot, often by getting bigger. But that's a losing battle that just makes the pump less and less efficient over time.
Okay, so that's the problem. And that, of course, brings us to the promise of regeneration. For about 2 decades now, there's been this absolutely revolutionary idea that has captivated researchers: the big what if. What if we could actually introduce new, reparative cells right into that damaged area to help it heal? The theory is that these cells could do all sorts of amazing things, maybe form new muscle, create new blood vessels, or, as we're now starting to think, release these powerful signals that just encourage the tissue around them to work better.
And that's the billion-dollar question, isn't it? It's such a simple and powerful idea on the surface. Can we use living cells as a form of medicine, delivering them right to the scene of the crime to jumpstart a healing process the body just can't manage on its own? It's a question that has launched an entire field of research. But, as you know, simple questions in medicine very rarely have simple answers.
The journey to find that answer has been a complete roller coaster. The early 2000s were just full of this incredible, palpable excitement. But then, as the 2010s went on, a lot of small studies came back with results that were, well, all over the place. Some were positive, some were not, and enthusiasm really started to fade. But science is persistent. Researchers kept refining their techniques, and now, after 2 decades of data, we've finally reached a point where a really big analysis can sift through all that information and finally find the signal in all the noise. And if you find this kind of deep dive valuable for understanding complex medical topics, please just take a quick moment to like this, subscribe to Digi-Gold, and maybe share it with someone you think would be interested. It really helps us create more explainers just like this. Now let's get back to the evidence.
All right, let's put this evidence under the microscope. We're not looking at one small study anymore. We are looking at a meta-analysis, a really powerful statistical tool that pools all the data from many studies to get a much clearer, much more reliable picture of what's going on.
So what exactly is a meta-analysis? Well, you can think of it as a study of studies.
By combining all the results, you dramatically increase the total number of patients, which gives you the statistical power to spot effects, even small ones that a single trial might have missed. In medicine, a really well-done meta-analysis is considered one of the highest levels of evidence you can get. It's the gold standard. And the way they did it here is what gives this analysis its real muscle. The researchers combined data from 18 different studies, which included over 1,700 patients. But here's the crucial part. They used a hybrid approach. For 8 of those studies, they got the raw individual patient data, or IPD. And that's a huge deal because it means you can dig into the specifics of every single person, not just the averages. They then merged that with the summary data from 10 other published trials. This kind of rigor gives us a very high degree of confidence in whatever we find.
Okay, so whenever I look at a new treatment, I strip away all the hype, all the noise, and I ask 2 fundamental questions, always in the same order. First, and most importantly, is it safe for my patient? And only if the answer to that question is a confident yes, do I move on to question number 2, does it actually work? So that's the framework we're going to use right now. Safety first, then efficacy.
So let's start with that all-important first question. I mean, think about it. We're talking about injecting living cells directly into the damaged hearts of very sick patients. The procedure itself carries risk. The cells themselves could cause problems. So before we even begin to think about whether this helps, we have to be absolutely sure it doesn't harm. And the main safety measurement in this analysis was the most important one of all: death from any cause.
And frankly, the result here is pretty striking. All-cause mortality in the group that got the cell therapy was 8.9%. In the control group who got the standard care, it was 15.1%. And that p-value of less than 0.001? To put that in plain English, there's less than a 1 in 1,000 chance this difference happened by a random fluke. So our very first safety signal isn't just reassuring, it's actually pointing toward a real survival benefit.
Okay, but what about other bad stuff? Let's look at MACE. That stands for Major Adverse Cardiac Events. It's basically a catch-all for the big 3: death, heart attack, or stroke. And here again, we see a huge benefit. The MACE rate dropped from almost 30% in the control group down to 18.4%. And look at hospitalization, which is so important for a patient's quality of life and is a massive driver of healthcare costs. A very similar, significant reduction. The data is just crystal clear here. From every major safety angle, this therapy looks beneficial.
All right, so we've cleared a huge hurdle. It seems safe. In fact, it seems beneficial. Now we get to ask the second big question. Does it actually work? Does this therapy do what it's supposed to do and improve the heart's function? For this, we look at objective measurements, and the main one was the change in that left ventricular ejection fraction we talked about earlier.
And the final number, after all the analysis, was a positive 2.51%. That was the average improvement in ejection fraction in the cell therapy group compared to the control group. Now, I know what you might be thinking. 2.5%? That doesn't sound like a lot. But in cardiology, you have to look closer. And this is where context is everything. First off, that 2.51% improvement was statistically significant. It is a real effect, not a random finding. But second, and this is maybe even more important, look what it did to the average patient's heart function. The average EF in the cell therapy group ended up at 36.1%. Now that number, 35%, is sort of a magic number in our field. It's a critical threshold we use to decide whether a patient needs an implantable defibrillator to prevent sudden death. Pushing a patient up over that line can have massive implications for their entire treatment plan.
Now let's look at another measure, end-systolic volume, or ESV. This is just the amount of blood left inside the heart after it squeezes. See, in a failing heart, the main pumping chamber tends to get bigger, stretchier, more baggy over time. And that's a bad thing. It's called adverse remodeling. What we see here is a complete reversal of that trend. The cell therapy group saw their hearts get a little bit smaller and more efficient. In stark contrast, the control group's hearts continued down that bad path, getting larger. This is hard evidence of positive cardiac remodeling.
Okay, so the big picture looks promising, right? It's safe and there's a real measurable benefit. But here's where this study gets really powerful, thanks to all that individual patient data we talked about. We can dig deeper and ask, do certain types of patients benefit more than others? This is how we move from a one-size-fits-all therapy to something much more targeted.
And the biggest headline from that deep dive is this: the greatest benefit was seen in male patients who also had that classic collection of risk factors for heart disease, things like diabetes, high blood pressure, and high cholesterol. That is a massive clue. Let's just break that down for a second. When we look at that positive remodeling effect, that reduction in the heart size, the benefit in male patients was highly significant. While the control group of men saw their hearts get bigger, the men who got cell therapy saw their hearts get smaller. It's a very clear signal. Now, the question we can't answer yet is why. We don't know. But it's a fascinating lead for future research.
And when we look at those risk factors, the picture gets even sharper. Patients with diabetes? Yep, they saw a significant benefit in that heart remodeling. Hypertension? Same story. And for those with high cholesterol or hyperlipidemia? You bet. It really suggests that the therapy is working particularly well in the very patients who have the highest burden of classic cardiovascular disease.
And this next finding is fascinating because it seems like the sicker the heart is when you start, the more it actually benefits. The analysis showed that patients starting with an ejection fraction of 45% or less saw that really significant positive remodeling. Even more, there was a direct correlation. The lower a patient's squeeze power was to begin with, the greater their improvement tended to be. It kind of makes sense. A weaker heart just has more room to improve, and it's absolutely vital for helping us target this therapy to the patients who need it the most.
Okay, one last detail, and it's a bit technical, but it's incredibly important. It turns out it's not just what you deliver, but how you deliver it. Most of these studies used a really sophisticated 3D mapping system, almost like a GPS for the heart, that lets you guide the injections with pinpoint accuracy. One of the large trials, though, used a more standard imaging system, and it saw a much higher rate of complications during the procedure. The takeaway here is crucial. The safety and the benefit might be tied just as much to the delivery technology as to the cells themselves.
Wow. Okay. We have covered a ton of ground here, from safety to efficacy to finding the ideal patient. As we get ready to wrap this up, I'd really love to hear what you think. What part of this analysis was most surprising or interesting to you? Let me know down in the comments below, and please, don't forget to like and subscribe to Digi-Gold.
So let's just wrap this all up. What did we find? A highly significant reduction in mortality and major adverse events, a significant drop in hospitalizations, and on the efficacy side, a real, even if it's modest, improvement in the heart squeeze power, which is backed up by strong evidence of positive remodeling. The picture is very consistent, and it's all pointing in one direction.
So we finally arrive at the verdict, and I think the study's own authors put it perfectly. The improvement is clinically non-robust. That means a 2.5% bump in EF isn't going to dramatically transform a patient overnight. However, it is a statistically significant improvement. The effect is real, it's not a fluke, and it comes along with very real reductions in death and hospitalization. Look, this isn't a cure, it's not a home run, but you know what it is? It's a solid hit. It gets us on base. It's a real, measurable step forward. Now, what's really exciting is what this all means for the future. We know something good is happening, but the how and why is still up for debate. The old idea that these cells were turning into new heart muscle, that's mostly been set aside. Now we think it's all about something called the paracrine effect. And this quote introduces this really cool idea called the hit-and-run mode. Think of it like this. The cells don't have to move in and build a new house. They just have to show up, deliver a package of instructions that tells the native tissue how to heal itself, and then they can leave.
And so all of this leaves us with one big, final question for the future. We have a clear signal. We have real clues about which patients benefit the most. We know that the delivery technology matters. We even have a good theory for how it all works. So the question is, if we can build on this foundation, if we can perfectly match the right cell type to the right patient delivered with the right technology, could this approach evolve from a modest benefit into a truly transformative cornerstone therapy for repairing the human heart? Based on this evidence, the answer feels like a very hopeful yes. Thank you for joining me.